Deep neural networks are being applied to a wide variety of regulatory genomic tasks, in most cases demonstrating superior performance compared to previous methods. Despite their promise, a major roadblock to their widespread adoption is their low interpretability. Convolutional neural networks are the models of choice as convolutions can be intuitively thought of as a position-weight matrix, or PWM model, and hence, a CNN can be considered a hierarchical generalization of PWMs. It was previously thought that first-layer filters learn motifs and deeper layers build upon these to learn motif interactions, and so it is common practice to visualize first-layer filters to demonstrate that the filters learn representations that resemble known motifs. However, these filters are seemingly cherry-picked, as the vast majority of filters don't seem to learn motifs in practice. Recently, the COO lab has shown that architectural choices significantly influence the extent that first-layer filters learn representations of motifs. And so, visualizing the filters of a CNN only makes sense when they're intentionally designed to do so. Hybrid CNN recurrent neural network architectures have recently emerged, in a few cases improving performance versus a pure CNN model. Many of these hybrid models still validate that their network has learned interesting biology by visualizing the first-layer filters. However, it remains unclear whether this makes sense. What are the design principles that lead to motif representations in first-layer filters? Previous research has illustrated that properties like max pooling size influence the extent that sequence motif representations are learned by first-layer filters. Here we conducted a small-scale study that explores the role of max pooling size and activation functions on model interpretability within a hybrid CNN bidirectional LSTM model, similar to popular state-of-the-art architectures such as DanQ. Here is our design of the hybrid network. The first layer is a convolutional layer, followed by a max pooling layer, a bidirectional LSTM layer, max pool layer, and a fully connected hidden layer. Using this model as a base model, we explored different max pooling sizes while keeping the rest of the network fixed. We call our model hybrid 50 if we use a max pool size of 50 after the first layer. We also explored how the representations of these models change when switching the ReLU activations to exponential activations in the first layer, which is a recent design principle developed by the COO lab that greatly improves motif representations in CNNs. To be able to evaluate the model's motif learning ability, we created a synthetic dataset that is an overly simplistic view of TF binding. Each synthetic sequence is 200 nucleotides long and implanted with 1 to 5 known motifs selected from a pool of 12 TFs, and the model simultaneously makes 12 binary predictions for their presence in the sequence. Although we are not using actual biological data, this allows us to accurately measure the model's power to detect TF motifs in ideal conditions. All models are trained with the same protocol. After training, we extract the first convolutional layer filters, visualize them as sequence logos, and compare them with the ground truth motifs. A comparison of the filters of hybrid 50 and hybrid 4 showed that more motifs were learned by the hybrid models with a larger max pooling size. For instance, 23 out of the 32 of hybrid 50's filters successfully learned ground truth motifs, while hybrid 4 only had 3 filter matches. Since the performance of hybrid 4 is better than the hybrid 50 model, this suggests that the bidirectional LSTM layer is assembling whole motifs from the partial motifs learned in the first layer. This trend is consistent with the design principles we observed in CNNs. Interestingly, if we change the activation function of hybrid 4 from ReLU to an exponential, nearly all of the filters now learn ground truth motifs. As shown here, the motif representations are much more clear with exponential activations. The number of motifs learned increased from 3 to 25 out of 32 filters, and the motifs visually match ground truth motifs much better. This trick was previously demonstrated for CNNs, and now we can say that it extends to hybrid CNN-RNN architectures. We also explored other architecture choices that didn't influence the first layer filters much, such as the number of units in the bidirectional LSTM and the first layer filter size. This study shows that similar to CNNs, representation learning of sequence motifs for hybrid CNN-RNNs are highly sensitive to architecture choices. Thank you.